All right, hi, welcome everybody. Good morning, good afternoon, wherever you are. I'm Christopher Dent. I'm a manager at AOAC, and I'm also the CASP program lead. And I'd like to welcome you to day 3 of the first series of the CASP Training and Education Working Groups webinars. I'm just going to go through a few very quick basic things, and then I'll turn things over to the presenters. So first off, a very big thank you, as always, to our CASP advisory panel members. Without the advisory panel members and their support, CASP would not be possible. So all of them are listed on the screen right now. If you're not up there and you'd like to be, if you'd like to help drive the direction of CASP going forward, please go ahead and contact Alicia Michael John, our Director of Business Development. Her email and phone number are on the screen right now. If you don't have time to write all that down, this recording will be provided to you later. Couple reminders. Mid-year meeting is coming up really fast. It's going to be held virtually from March 15th to March 19th. The CASP meeting is a part of that, and it'll be held on Wednesday, March 17th at 1 p.m. Eastern. It's going to be a good meeting. We're going to get updates from all 5 of the working groups. We're going to get some general program updates, discussion on some next steps, as well as a special guest speaker. That agenda will be posted on our website, aoac.org, pretty soon, and that's where you'll want to go to register as well. So a lot of our CASP members aren't aware that you can also become an individual member of AOAC. It's pretty affordable, and you get a lot of really great benefits out of it. I'm just going to flash them up on the screen quick. You get discounts to OMA, discounts to our meetings, a few free magazines and journals, things like that. More information about that also at our website, aoac.org. So housekeeping items for today's session. While the presenters are presenting, please feel free to type questions into the chat box. After the presentations are done, the working group chair, Toby Astle, he'll moderate a Q&A session reviewing all those questions that you've dropped in there. During the presentations, please be sure to keep your video off and your audio muted. Finally, a survey, as well as the recording, will be sent out to all registrants after the event. That's enough from me. If you just give me a moment, we'll get those presentations fired up.
Good afternoon. My name is Toby Astle, and as the chair of the CASP Education and Training Working Group, it's a pleasure to bring today's session to you. Today we'll be focusing on the microbial contaminants found in cannabis, and we have 4 excellent industry speakers who will be each sharing their insight into this topic. Starting, we have Michael Esposito from MCR Labs. Michael will discuss the past, present, and future of microbial testing, which technologies are suitable for your lab as they try to test for various pathogens and microbial species. Following Michael, Alex Tudor from TEQ Analytical Labs will discuss the challenges faced in the cannabis micro lab. Following Alex, Michael Lowenstein from Q Laboratories will discuss the topic of expanding into the world of cannabis testing, lessons learned from an established food and pharmaceuticals laboratory. And finally, Pat Bird of PMB Biotech Consulting will cover the topic of, is cannabis legal in my state? Is it safe to consume? And speak to AOAC's role in developing standards, educational guidance, and certifying methods for microbial analysis. Please sit back and relax, and please enter any questions you have into the chat box. After the 4 sessions, there will be a live Q&A with the panel. Thank you and enjoy.
All right. Hello, everybody, and thank you for joining us today. My name is Mike Esposito. I'm a microbiologist over at MCR Labs, and today we're going to spend some time talking about microbiological testing methods. First, we're going to go over what your basic objectives are when you're running microbiological testing at your lab. So primarily, we're doing both the detection of target organisms and the quantitation of larger organismal groups. For the detection of target organisms, we're mostly looking at pathogenic organisms, things that could cause foodborne illness, etc. And these are typically recorded as a presence or absence within a certain massive sample. These are really critical because if we miss anything on these pass-fail tests, there could be really dangerous medical implications down the line. The other type of microbiological testing that you might be doing would be quantitative measurement of organismal groups. This is typically more of a quality indicator, kind of gives you an idea of what the background of your product has been. Usually it's broken down into 4 larger subcategories. That would be your total aerobic count, which should encompass everything. Off to the side, you would have total yeast and mold. And then again, within your aerobic count, you would have your enterobacters and your total coliform units. And then sometimes within that, you could be looking for fecal coliforms. The enterobacters and the total coliforms are more associated with those pathogenic organisms that we discussed a slide back. So those are really the ones that you want to be watching out for. And these are typically reported in units of CFU per gram, which is a colony forming unit. All right, so there are a few classical methods that we use for these testing. The first would be plating, which is the oldest of the methods. This has been used since the late 1800s, and it involves you taking a little bit of a sample, running it through some sort of liquid to get it homogenized, and then actually directly applying it to something like an agar plate, which would have some growth media or food for those organisms to eat. Then you would allow that to incubate for a certain amount of time, and you would be physically counting the colony forming units on your plate there. In order to get to those more specific groups that you're trying to quantitate, you can use selective medias, which would either involve a food source that only a certain class of organism could eat, or something potentially hazardous to the organisms you don't want to measure, like an antibiotic. All right, the next evolution of the culture-based test would be the most probable number. Now, this is a way for you to generate CFUs by getting an MPN, or most probable number result. The principle behind this is what's known as extinction dilution. So you're hoping to dilute your sample stepwise to the point where some of the tubes or individual vessels you're culturing in are going to have absolutely no organisms in them. So essentially, you'd be running your stepwise dilutions, incubating all of your tubes or whatever vessels you might be using. And then at the end of that incubation period, your vessels will have either a growth or no growth binary state. And now, based on how many of those tubes at the different dilution or volume levels have growth or non-growth, you're able to algorithmically determine your MPN value. And an MPN essentially correlates to a CFU. These also can use selective media, just like the plating methods we talked about earlier, and that's how you're able to kind of break it down into those subcategories. The last quantitative method, classical, that we're going to talk about is cytometry, which frankly, I don't see a lot of people using in high throughput labs. It takes pretty high level of skill, but this involves you physically counting the organisms on your sample using something like a microscope, a slide that has a grid pattern on it known as a hemocytometer. And it's really hard to differentiate between the organisms unless you're an incredibly well-trained microbiologist. You might require some dyes in order for you to determine if a cell is living or dead, but this doesn't actually require any incubation time. It's just an extremely difficult technique. All right, so now we're going to look at some of the challenges to these classical methods. First of all is the metabolic variance between microorganisms. Each organism is not going to have the exact same perfect culture conditions, so it's not so much one temperature is perfect for all organisms, as much as this is the temperature that on average is going to be best for this class of organisms. You can see in this graph to the right, there's some organisms that will have an optimal incubation at 90 degrees Celsius, and some that are all the way back down to 10 degrees Celsius. Aside from the temperature variation, there's also different preferences for different food sources between organisms. And additionally, if one organism is going to outperform another on a plate, it could easily overtake that and kind of obscure it and make it unmeasurable. The last big problem with metabolic variance is that some organisms actually require a living host in order to grow. So things like a powdery mildew on a plant is not something that you'd ever be able to quantify using a culture-based method, since it really requires that living plant host in order for it to proliferate. All right, another challenge to this is drug-resistant organisms. So earlier we talked about selective medias that require antibiotics in order for you to kind of filter out the things you don't want to grow. But unfortunately, as we see in the medical industry and the agricultural industry, it's very possible for microbes to develop drug resistance genes. So you can see in the image to the right, there's a plate that has some organisms growing in it. All of those individual white circles represent an antibiotic. And as you can see, some of those antibiotics are extremely effective there's a large halo zone of inhibition there that the organisms can't grow in, but some of them are much less effective. You see they're able to grow all the way up to the actual antibiotic. All right, and then lastly, there's the structural issue of the variation between microbes. You can see in the image to the right, some largely filamentous fungi are extremely tough to count on a plate. They'll just make a large web of hyphae, and at this point, you can't really determine if you have one CFU or if there's 20 in there, and they just kind of all clump together. If you're working with bacteria and you have far too many on your plate, you run into a TNTC, or too numerous to count result, and that means you're going to have to reprep your sample at a much higher dilution factor in order for you to actually physically see how many are on that plate. Then you just multiply back there to get your true number. All right, so now we'll talk about some of the modern methods. Most of these are molecular methods, meaning that you're utilizing the DNA of an organism to detect or attempt to quantify. So first we'll talk about quantitative PCR. PCR is known as the polymerase chain reaction, and this is how we're actually able to take a target segment of DNA and using specific primers to that segment of DNA and enzymes known as polymerases, we're able to artificially reproduce that specific strand of DNA over and over, creating what's known as amplicons. The quantitative aspect of this PCR is when you use a fluorophore during your PCR reaction, and that's going to actually release a fluorescent response every time the amplification reaction occurs, and that's something that's measurable by our instruments. As you can see to the right, the curve we're looking at here is actually the fluorescence response, and it kind of shows you going through the cycles, the process of PCR. So starting out at cycles 2 through 14, you have relatively low amplification, but once you hit around 18, you start to see a sharp upswing on the curve there, and that indicates to you that you've reached the exponential growth phase. So you're really just producing tremendous amounts of amplicons until you get around cycle 42 or so, where it starts to flatten out, and that represents you kind of expending all of your reagents within your reaction well. You see something called a CT or a CQ value. On this image, it's a CT value equals 22. That is the point at which the fluorescence response crosses an established threshold. And that's what you kind of utilize if you're going to use this for any sort of quantitative measure. A earlier CT value indicates that you had more of your starting DNA template in your well, allowing you to hit that exponential growth phase earlier, versus a later CT value, which could indicate maybe you don't have as much target template DNA in there, so you're going to hit a few more reactions before you really hit that exponential growth phase there. So more or less, a lower cycle means you have more template DNA in your reaction. A higher cycle means there's less template DNA. All right, next is the microarray, which is another evolution of the PCR. Rather than relying on that fluorescence curve response here, you are going to be amplifying your PCR product, getting as much of those genes as possible, and then you're going to apply that product to a microarray chip. And what these are chips that are completely covered in these oligonucleotides. Earlier we talked about primers. These oligos are synthetic strands of DNA that are specific to a target gene. So if you're applying your DNA amplicons that you generated through PCR to this microarray chip, they're going to bind to the available sites, and then through the fluorescence of those DNA amplicons on those binding sites on your microarray chip, you're able to get an idea of how much starting DNA you had to begin with. All right, next is the digital PCR, which is the most recent evolution of the PCR. Digital PCR, rather than you using one well and you're taking a measured response curve out of that, you're breaking down your reaction into thousands or millions of individual PCR reactions. And each of those reactions is going to have a binary state of amplified or non-amplified. The droplet digital PCR is something that you can see in this image to the right here. There's sort of like an emulsification of oils. And within each mini bead of oil there, you're going to have your reaction, which is either going to be amplified or non-amplified. And like the other methods, this kind of gives you an idea of how much starting template DNA you had. All right, so there are some issues when we're using these technologies for different applications. These molecular assays are really perfect for your pathogenic presence or absence tests because they're not relying on any quantitation. But once you try to extrapolate a CFU value out of these CQ values, you kind of run into some issues. One of the primary issues you run into is the extraction efficiency. So in order for you to even run your PCR reaction, you're going to need to first liberate that target DNA. And there's not really a one-size-fits-all method to do this. Different organisms have very different physiologies, cell wall thickness, etc. And as you can see at this image to the right, something that might generate an incredibly high amount of yeast DNA is going to be completely ineffective against an aspergillus potentially. You can see here the yeast lysis method of extraction for Candida albicans resulted in 12,980 picograms per milliliter of DNA versus the same approach used on Aspergillus fumigatus, which yielded these people 0 picograms per milliliter. And you'll see with the more thick-walled organisms, you really need to get aggressive. You might require bead bashing and high temperatures, which could introduce inhibition in a different, less hard-walled organism. All right. The next challenge here is the question of enrichment, which is essentially taking your sample and putting it into a growth media and allowing it to incubate for a brief amount of time before running your molecular assay. For most of these presence absence, it's really critical for you to have enrichment here. You want to be sure that if there's one dangerous E. coli organism in your sample, it has had the chance to multiply to the point where you're going to be 100% confident in detection with your molecular method. You can see this example to the right was, I believe, a bunch of cattle carcasses that they ran. It was 647 total samples. They ran a post-enrichment and a pre-enrichment assay on each of these samples. And the pre-enriched samples only had 128 out of 647 positive hits versus the post-enrichment more accurate results, which were 614 out of 647. So this enrichment is really critical in any sort of detection of a pathogenic organism. All right, the last huge issue with molecular approaches for quantitation is the issue of copy number variation. So you'll notice that I've only been talking about the quantitation of template DNA rather than the quantitation of organisms. And this is because these molecular methods really are just giving you an idea of how much starting DNA there was, which doesn't necessarily correlate one-to-one with how much of the starting organism there was. A lot of these very closely conserved genes that we're using for quantitation, it's extremely variable how many copies of that gene are within an organism. This varies across species, and even within species, there can be a huge variation. This image to the right shows a study of the rDNA copy number for various isolates of, I believe, Aspergillus fumigatus. You can see the organism with the most copies of that was somewhere around 65 gene copies of one target versus one that had 5 copies of that same target. So you're looking at a 13-fold difference there. And when you're trying to generate a CQ to CFU conversion, the way you could go about that would be to develop a standard curve there to try to correlate it. But if you're building your standard curve off of a 65 copy organism and you end up running a 5 copy organism, you might end up underreporting by a factor of 13 or something like that. So it's very tough to get an accurate idea of how many organisms there were based off of the starting DNA template count you had. All right, so all things considered, when you are planning your microbiological approach at your lab, you really want to be sure that you're 100% confident in your detection. These molecular methods are perfect for that. You're able to specifically design primers for a pathogenicity gene. For instance, the STX2A gene in this Shiga toxin producing E. coli organism to the right. You're able to, using that, not fail any of your samples for the more benign E. coli that are really common in the gut. And because you're not trying to generate a CFU value here, you're not so much concerned as to what your CQ represents. You're just knowing that, oh, we have a CQ value here. We've enriched our sample. There is dangerous pathogenic material in here. Or we've enriched fully. If there was anything in there, we would detect it. And we haven't, so it's a pass. And then lastly, for quantitation, you want to provide as accurate of a result as you possibly can for your client. We know in microbiology, you're never going to be as precise as you are with analytical chemistry, but we know that we have existing technologies that have been used for decades that are, extremely robust. There's trusted methods available through like the FDA Bacterial Analytical Manual or the United States Pharmacopoeia, et cetera. And even though it's incredibly appealing to look at these new molecular methods to produce quantitative results, looking at the throughput you can offer your client, a 1-day turnaround versus a 3 to 4 day turnaround, we just need to be mindful that we're not really getting as precise as we could with our CFU values there. All right, so looking forward, hopefully we're going to find a point where we can combine the increased throughput with the increased accuracy. Maybe down the line, we could be looking at something like a combination of the MPN assay with the qPCR assay, where we're able to get sort of a volumetric or a dilution-based idea of how many organisms are present, and then rapidly amplify DNA for detection. But at this point, we're not quite there yet, and these are all things that are coming down the line. So anytime we're adopting new technologies into our lab, we want to be sure we're extremely robust with our validations of these technologies. We're getting as much literature as possible, and we're really just verifying that the results that we're offering to our clients are as accurate as they possibly can be. And in order to do that, you might be needing to use a combination of multiple technologies in your lab for now. All right, that's all I have for you guys today. Thank you very much for spending some time to go over the microbiological methods with me.
Hello, everyone. Thank you so much for joining us today. My name is Alex Tudor. I'm the Director of Business Development at a cannabis testing laboratory just outside of Denver, Colorado, called Tech Analytical Labs. We are located on the University of Colorado Anschutz Medical Campus. My background is in food microbiology. So today I will be talking about some of the challenges that we have faced as a cannabis testing facility from the lens of a microbiologist. My experience is from a lab in Colorado. So what I speak on today will be specific to what we have seen in our state, but other states or countries may encounter some of these challenges, but also may encounter others. So Tech was founded in 2014, right at the beginning of the legal cannabis market in Colorado. Tech Analytical Labs was founded as an offshoot from a ISO 17025 accredited food and drug testing laboratory with over 70 years of experience called Industrial Labs. Tech was the first ISO 17025 accredited cannabis lab in Colorado and the fourth in the US. And like I mentioned, we are located on the University of Colorado Anschutz Medical Campus. So it's hard to nail down exactly what challenges a cannabis testing lab will face, but I will highlight from our perspective and experiences, challenges related to limited sample sizes, a lack of reference or compendial methods to draw from, as well as unique matrix and client-specific challenges. So to begin the discussion today, I'll get into some of the challenges faced with cannabis matrices in a testing lab. In regulatory testing for cannabis, many growers and producers are unwilling to submit sample sizes that would be considered sufficient in the food testing industry. Typical food industry samples can range from 25 grams up to 375 grams. So these types of methods that we can use as reference aren't validated to the smaller sample sizes that we see. The testing requirements in Colorado are not seen as necessarily beneficial or valuable to cannabis growers and producers, but instead burdensome, inconvenient, costly. There's pressure put on labs to validate methods on the smallest sample sizes possible. And in most cases in Colorado for microbial testing, that sample size is 1 gram. So state regulations require a specific sample size be submitted based on total production lot. But what has been missed here is that one sample submitted is then analyzed for multiple tests, thereby decreasing the available sample for analysis. For example, a 4-gram sample is required for harvest batches of flour weighing up to 10 pounds. So that amounts to just over 1% of the total batch or lot produced. Additionally, if a 4-gram sample is submitted to the testing lab, part of that sample may need to be subsampled for chemical testing like potency, pesticides, terpenes, or residual solvents. So we're not working with a lot of material to test. And if we actually get to test maybe 1 gram of the 10-pound lot, that ends up being 0.02% of that batch. So the likelihood of finding a positive in that is relatively negligible. And in Colorado, the producers are responsible for their own sample collection, which poses additional challenges. Regulations define a 4-gram sample as 8 individual half-gram samples to total 4 grams. And some clients either do not know this or intentionally avoid it, and it falls on the lab to enforce this regulation. It also falls on the lab to ensure that the samples we receive don't appear to be adulterated, which can put the lab in a pretty precarious position. So next, I'm going to touch on the lack of reference methods available to us in the cannabis industry. So before AOAC stepped in with SMPRs and a team of scientists who want to advance and improve the cannabis testing landscape, cannabis testing labs were largely on their own in regard to validating test methods. So many test kit manufacturers had not validated their test kits on cannabis matrices, so internal work had to be performed to ensure the methods would be fit for use with the different test matrices that we see at the sample sizes that were outlined by the testing regulations. So to some degree, testing labs were sort of on their own in defining which methods they would want to validate and then use based on the expectations their own clients had. There was, and there still is, although this is changing, limited support for the labs in that case. In many other industries, there's often a big bad wolf type of regulating body that holds labs and producers and manufacturers to a high standard, but also can be utilized as a resource and for guidance in certain circumstances. The cannabis market doesn't yet have this type of beacon, but AOAC is well poised, especially as more SMPRs come out, to become that resource for us. We know that cannabis and its byproducts are unique and pose unique challenges, but using pre-existing methods for matrices in other industries that may be similar would be a great starting point. This, at the very least, could provide insight into testing methods based on matrix, while still recognizing that cannabis will require its own unique methodologies. At this point, all matrices are tested using the same methods, but that may not be the most accurate way to analyze them. How do you build uniformity in the lab with so many different matrices? Additionally, there are some analytes that use the same method for screening and confirmation. As the science progresses, this will most certainly change, but with the limited resources in methods of analysis with this emerging industry, this is sometimes the case. And because there were not standard methods defined by regulators or even a standard validation process defined, there's been inconsistencies in labs' validation methods and robustness. It wasn't until 2 years ago that the state regulations in Colorado required ISO accreditation. So before that, the variability in quality was pretty dramatic. Also, many of us see the value in audits performed by A2LA or others, but some in the cannabis space view audits as inconvenient or unhelpful. Variation in quality systems is an interesting thing to consider here as well. Things like measurement uncertainty or the stringency of a lab's quality parameter may result in a higher number of failures, which could frustrate clients. Where is our incentive to provide a more accurate test when failures based on QC push clients away? So matrix specific challenges are a pretty interesting component. So I alluded previously to clients feeling that testing was an inconvenience. Additionally, the value of their products is defined in large part by the potency results that labs produce. In Colorado, test samples are prepared by representatives of the cannabis grower or producer and not by an outside party. So this, as you can imagine, can lead potentially to manipulation of samples, both to increase potency and decrease microbial counts. So it's up to us as the testing lab to identify when this happens, which, like I mentioned previously, puts us in a really weird position. We're here to help and support our clients, but if someone is trying to manipulate their samples and doesn't necessarily see the value in the high quality science that we're trying to produce, it might mean that we're not the best fit for that specific client. In the food industry, samples are often presented to the lab in their final, ready for customer packaging. In cannabis, this is not always the case. We'll often have samples submitted in Ziploc bags, sandwich baggies, or other packaging types that may not have been prepared in an aseptic fashion. Sample labels from clients are sometimes in the package with the test sample itself. Other times, labels are stapled through with a plastic bag, piercing the bag and potentially contaminating the sample before it even gets to the lab. We see a wide variety of matrix types at the lab. Dried cannabis flower is very different from an extracted oil or concentrate, which is different still from a hybrid matrix or an edible matrix. Each matrix type requires its own internal method verification, and different matrix types may pose unique challenges to adequate analysis. Yeast and mold is ubiquitous in many crop type commodities, and cannabis flower is no exception here. The way that cannabis plants are grown and then are dried oftentimes results in inhomogeneity, and even within the same plant. So there are parts of the plant that are much more prone to higher mold counts than others. So when two testing labs perform a quantitative yeast and mold analysis on samples from the same harvest batch, oftentimes the counts can be wildly different. This can be something that is difficult to explain to a client and difficult for a client to understand and comprehend. So to give you guys a more clear understanding of the actual diversity of sample types we see at the lab, this is an example of cannabis flower or bud sent for analysis. More examples of flower submitted. These actually are in roll-pack bags, which is something that we try to push our clients to use just to protect the integrity of their sample a bit better. These photos show how flower is dried and cured before analysis and sale. So plants hanging kind of just upside down in drying rooms on wire racks, kind of just to dry and be ready for sale. This is an example of a concentrate on the left-hand side, actually a distillate kind of concentrate, and then a wax concentrate on the right-hand side. These are pictures of what we call shatter and sugar. More concentrate, similar but different, all extracted cannabinoids from the cannabis plant. And then this is diamonds and sauce, which is also a concentrate type. So you can see those crystals in there are actually THCA crystals, and that liquid is a terpene-heavy kind of sauce. We also often see combined matrix types into a single sample. So this is concentrate combined with flower. So you can kind of see the different examples of that. And then with this, the questions that you have to ask are, what do you do with validation? Is a new validation necessary, or is the independent matrix validation sufficient with this hybrid type of sample matrix? This is an example, these are two examples, I guess, of edibles and specifically edibles with large inclusions. So how does that impact sample size, analysis, validation of the method used? So now I'm going to get into a little bit of the client-specific challenges. Unfortunately, testing labs in the cannabis industry are not seen as value-add resources, like I've kind of alluded to, or a way to support and protect a business. The lab is often the bad guy in reporting failed test results or unexpected test results. In many cases, the immediate reaction of our clients is that our science, our methods, our analysts are at fault. While we understand that there are mistakes that can be made, it can be frustrating to feel that the science has to be defended at every turn. With this, I'm saying that you need to have a thick skin and a strong quality system to fall back on. The testing lab is often seen as the mechanism to move products to sale, which is that fulfillment of the check the box mentality of testing that we see. Occasionally, if we provide a result that is unexpected or unpleasant, some clients are just not open to a conversation about it, and instead will change testing labs to get results that they like better. A lack of understanding from our clients of the science and methods used is especially troubling when discussing turnaround times with them. So in Colorado, the cannabis testing industry has largely adopted rapid yeast and mold Petri film due to the short time to result. In many other industries, yeast and mold quantification takes 5 to 10 days or more, but in cannabis, yeast and mold turnaround time is only 3 days. Many times, cannabis grow facilities are indoors and the plants need a warm, humid environment to grow. The plants are often on plywood tables up off the ground, and to put it politely, there is a lot of opportunity for improvement in facility cleanliness and microbial contamination awareness in the cannabis industry. We know the expectations of FDA, HARP-C, GMP facilities are vastly different, and we know the Standards and Policy Working Group is trying to drive GMP implementation, which would be an amazing start. So what are we missing, right? Are we screening for the appropriate microorganisms? What are we not looking for? How accurate are our methods based on the matrices and sample types that we see? Some of the challenges that we know exist in the facilities where these cannabis plants are grown, and how can we combat and help with that? Implementation of rules is an interesting thought here, as a lot of the regulations were made maybe by lawmakers and not necessarily scientists or people familiar with the cannabis space. So I think there's a lot of opportunity there. And then in educating regulators, like what we're trying to do today in terms of what the challenges are in the lab, how we maybe can combat those. But there will be challenges within this industry for years to come. So as long as we can kind of put our heads together and think about what we can do to combat those, I think we'll be in great shape. If another industry is doing something, why aren't we doing it? We can always look to our friends and colleagues in other industries for guidance and as a resource. So thank you so much. Please feel free to reach out if you have any questions or further concerns or want to discuss anything. And thank you so much for joining us today.
Hello, everybody. My name is Michael Lowenstein. I'm the Vice President of Scientific Consulting at Q Laboratories in Cincinnati, Ohio. Q Labs is an established food and pharmaceutical testing lab. We also service dietary supplements, a variety of industries, and have done for the past 55 years. Over the past several years, though, as this new cannabis industry has been booming, we have started getting into this cannabis testing space. And during that time, we've learned a series of valuable lessons that I'm going to try and convey to you today. One of these lessons is that there's this rapidly evolving regulatory landscape, right? There's the USDA, there's the DEA, the FDA, all of which have conflicting concerns. It's not always clear where one jurisdiction ends and the other one picks up. This is particularly true regarding CBD. So we've learned a lot about that over the last several years, and I'm hoping to share that with you today. That also feeds into, okay, what's a best practice and what's actually a requirement by law? There's, depending on jurisdiction and depending on regulatory body, and depending, quite frankly, on who you ask, you might get different answers. We've learned a lot about that as well. And then as a lab, you're going to have to make smart investments in technology and people to serve cannabis clients. And while this is a microbiology session, and all of these lessons certainly apply to a microbiology lab, they really apply to your lab as a whole. So if you have an analytical chemistry function or some kind of physical characterization function, these same lessons I think are valuable for those as well. With that, let's jump in. So who's the first regulatory body that we're concerned with when it comes to cannabis? And I would say that's the United States Department of Agriculture, the USDA, right? It was the USDA Agricultural Improvement Act of 2018, it has come to be known as the Farm Bill, right? That federally legalized hemp and hemp derivatives. Quick refresher on what's hemp as distinguished from marijuana. Both hemp and marijuana are cannabis. Hemp is cannabis that has a THC content of less than 0.3%. Marijuana has a THC content of above 0.3%. The USDA Agricultural Improvement Act of 2018, the Farm Bill, said hemp and the derivatives of hemp are federally legal. The AOAC CASP SMPR for potency is specifically mentioned in that Farm Bill. Why do I mention this? Well, because it sort of set a precedent that AOAC is deferred to as an expert body, as a standard setting body. And we can expect that the same kind of respect for AOAC's work in this space will apply for microbial contaminants and other SMPRs that come out of AOAC, and specifically CASP. Next, we have the Drug Enforcement Agency, right? The DEA. We said that less than 0.3% THC is hemp and is legal. Above 0.3% THC is marijuana, which at a federal level is still a Schedule I controlled substance and is illegal, right? So their enforcement hinges on what's the THC level in this product. Do you have to have a DEA license as a lab? That was one of the questions that we had ourselves as we were getting into this space. If you're planning on testing hemp biomass, so that's the actual raw flower or stalk, or any part of just the raw plant from the field as it's harvested from the field, the answer is yes, you have to have a DEA license. But what about if you're not interested in testing the agricultural material of hemp or more broadly cannabis, right? But you want to test instead the intermediates or the finished product. So whether that's an oil or a tincture or a topical of some kind, what then? Do you have to have a DEA license? What we've come to find out is technically the answer is no. But although you maybe don't technically have to have a DEA license, it's reassuring for everyone involved. It not only shows your client that you're very serious about servicing this industry, but let's say you get a finished product in and you test it for potency as a release product or a release criterion, and it tests above 0.3% THC. Are you in possession now of a Schedule I controlled substance? Technically, yes. And so what do? Right? If you didn't have your DEA license, you wouldn't necessarily have all of the procedures in place with reporting structures and all of that to deal with that material, and you just be in a tough spot. So what we have found is go ahead, get your DEA license, and in that instance, the DEA knows you're not trying to do anything shady. They know that if you get one of these products that tests hot, you can contact them. You'll have procedures in place for reverse distribution. Makes everybody much more comfortable. With an estimated 20% of hemp grown in the U.S. testing above 0.3% THC, this is a legitimate concern to have. Now, everybody's favorite topic, the FDA, the Food and Drug Administration. I'm going to talk about this mainly in the context of cannabidiol CBD, not only because it's sort of the flavor of the month, it's flavor of several years, I guess. It's the one everybody knows about. It's the most popular cannabinoid right now that we're seeing currently marketed, but also because it has the most to do with the FDA right now because CBD is an approved drug. Because it's an approved drug, it cannot be sold as a dietary supplement or added to foods. Approved drugs, there's an exclusionary rule that you can't sell a drug as a dietary supplement, and you can't add a drug to food. So technically those products right now are illegal. And then if you're making one of those products, a GMP or a CBD product, GMP controls will be expected for those products. And some of those things may be new if you're traditionally a food lab and you're not familiar with the varying degrees of good manufacturing practices. To sort of drive this point home, these are snippets from a report that was commissioned by Congress and issued by former FDA Commissioner Stephen Hahn, talking about exactly this topic. And I'd encourage you, I give you the source there at the bottom, I'd encourage you to read the entire thing. It's not very long. It's very easy to read. But specifically here, they're talking about dietary supplements. He says, okay, under current law, CBD products cannot lawfully be sold as dietary supplements. He goes on to say FDA has discretion to remove this exclusion through rulemaking. That's what lots of people are hoping that they will do, but they haven't done so yet. Likewise for food, right? It's not currently lawful to add CBD to human or animal foods. And he goes on to say, look, we have safety concerns about adding CBD to food products. So I'm hammering on this point a little bit because we hear all the time from our cannabis clients that, well, we're waiting on the FDA to speak, or the FDA hasn't been clear on what their expectations are for CBD products. The fact is the FDA has been extremely clear on what their expectations are. Again, what they're going to do in the future in terms of rulemaking, no one can say, right? We have to watch the market, and that's what the lab can be doing. The lab can help their clients sort of keep an eye on what the FDA seems to be doing. But right now, their position is clear. What does that mean? Well, all of these CBD products are in our supermarkets. There's more and more showing up every day. And so, fine, they're technically illegal, but all these companies aren't getting shut down. What's going on? Well, FDA is they're monitoring the market, and they're taking selective enforcement action. What is the key driver that's driving whether or not they're going to take enforcement action against the company? It all comes down to the claims that a company is making. If you're making wild health therapeutic claims about a cannabis product, it's going to draw the attention of the FDA, right? If you make health claims about any product, CBD or otherwise, then you're selling a drug. And those therapeutic health claims have to be substantiated with science. And right now, there's very limited data available to substantiate many of these claims that companies are trying to make. So if a client comes to you and says, oh, hey, I'm looking for a lab partner for my new cannabis product that is great, and we know that it treats Alzheimer's disease, or it treats nausea associated with cancer and things like that, alarm bell should be going off in your head. So as a final point on this, right? If you're testing CBD products, that's fine, but you should be familiar with what are the good manufacturing practices expectations of a lab when testing CBD products. And while today, these are requirements for CBD products, it's reasonable to expect that those same kind of expectations are going to be expected for new cannabinoids that come to market in the future. Why? Because the whole point of these cannabinoid products is to, as health stimulants, right? So as new ones come to market, very likely that people are going to be trying to sell them as dietary supplements with positive health effects. GMP is going to be, in that case, also required. Today, it's certainly a best practice, and we'll get into that in a second. I'll also mention AOAC is very well placed to apply lessons learned from the stakeholder panel on dietary supplements and other key working groups in the industry to help drive standardization and clarity in this area. AOAC works closely with the FDA and other regulatory bodies, as well as, of course, lots of cannabis companies and method developers and things like that to help sort of translate for all the different disparate portions of this industry to help drive cross-functional harmonization and collaboration. What can a lab do? Well, the lab can keep a close eye on FDA guidance documents that come out and keep a close eye on warning letters that come out to proactively update their own procedures, and then education of their clients is applicable. So we talked about, all right, good manufacturing practices as a requirement for CBD products, right? But really, they're always a best practice. So we can give one example from a micro lab, right? Let's talk about matrix-specific method validation and suitability is what our personal care and drug clients would call it. The food industry would call that method verification, right? The point of it is the same. Let's ensure that the method that we've chosen to assay this product for microbial contamination is in fact capable of recovering the microcontamination that is there, right? Sounds like it's always a good idea, right? And it is. It's a requirement for drugs, but it's scientifically always a best practice. I'll also mention ISO 16140 Part 3 was just published. I'm mentioning this because if you're not super familiar with how to design a matrix-specific verification study, this would be a great template to use. And again, the industry just requires education on the value of this, right? It's not particularly useful, I think, for us to argue so much about, is this a requirement or is this a best practice? Once you explain to a client, look, you're investing in doing microtesting, you understand the value of doing microtesting. Don't you also want to make sure that the money you're spending on this, the time and effort you're spending on this, is giving you data that's actually useful? Once they hear that, they're very willing to move forward with that. Another best practice requirement kind of example is ISO 17025 accreditation. In our experience at Q Labs, just like the food industry, ISO 17025 accreditation seems like it's going to be very important for cannabis companies as well. Beyond that, many accreditation bodies are coming out with cannabis lab programs that are, it's ISO 17025 accreditation, but they take a particular look at what are the special, unique concerns that cannabis labs specifically have. A2LA, as an example, has one of these programs. And I mention A2LA because A2LA is very involved with AOAC and CASP. I'll mention really quickly, AOAC is an expert body recognized by ILAC for method accreditation. And the initiatives under AOAC CASP are driven by A2LA and ISO 17025 accreditation standards. What does that mean? Well, the proficiency testing program that AOAC CASP is coming up with, by which labs can check their proficiency, right, is driven by the requirements outlined in ISO 17025. Same goes for method validation requirements, right? There are new method validation requirements in the 2017 version of the ISO 17025 standards. We here in CASP are using that as the basis for which to develop requirements for our own method validation, and so on. So becoming very familiar with the ISO 17025 standard and getting accredited to that standard is going to be certainly a best practice, if not a requirement. And finally, for established food and pharma labs like Q Labs, many of the tests that cannabis clients are going to need are going to be new. So not only investing in the instruments that requires, but also let's not forget the analysts that are required to execute those tests, interpret the results, and troubleshoot issues. As you've heard today and through other sessions, cannabis is a tricky matrix. We're running into issues with cannabis that we haven't seen in other matrices that we've used that we've seen in the food and pharma space. That's going to take investment and time. And again, while this is largely a chemistry concern, right, that when we think about investing in expensive instruments, historically that's meant buying a lot of chromatography equipment, lots of very expensive analytical chemistry instruments. It's also going to be a concern for micro labs in this space too, because the cannabis industry is a young industry, and because they're a young industry, they're much more open, in our experience, to exploring alternative micromethods. You know, the established historical pharmaceutical personal care companies, they want to see counts on a plate. That's what they know. That's what they're comfortable with. But much like the food industry has been more open to rapid methods, the cannabis industry is open to rapid methods as well. So things like PCR-based technologies, ELISA-based technologies, a lot of these rapid methods can give you a competitive advantage as a lab. But also a lot of these cannabis clients are educating themselves by being involved in organizations like AOAC to understand that these methods exist, and they're going to be demanding that labs employ them. So if that's new to you, something to consider as a capital expense investment area for your company. Thank you very much for your time. If you do have any questions, please stick around to the end. We have our live question and answer session. Otherwise, feel free to contact me by phone or email at the contact here. Thank you very much.
Good morning. My name is Pat Bird, and I'm the principal consultant at PMB Biotech Consulting. Today, I'm going to speak to the role that AOAC has in developing standards, validation, guidance, and educational resources for the cannabis community, with specific regards to the microbiology cannabis community. So my presentation today is going to focus on several key challenges that our community faces and how AOAC fills a role and serves a need within those. The first topic we're going to speak to is this non-existent federal framework for regulations for microbial contaminants. So when you look at this from a state-to-state point of view, you can see that in the absence of this federal framework, states have individually developed their own testing requirements that they've put into place. This slide shows the variety that we see out there from state to state and really makes it hard for a lab to really establish themselves in one area and potentially move to another state as well, as they may have to update their technology they're using, their SOPs, their quality system to meet the various regulations that we see. Please note that this overview is really a general discussion or general outline of what laboratories require or what states require, and we've grouped them together as best we can, although there are some variations within each of these groups. Another way to look at that is, from a standpoint of what states really require both pathogenic microbial contaminant testing, STAC, Salmonella, as well as contamination organisms. So your yeast and molds, aerobic counts, E. coli's, coliforms, versus those that just test for pathogens, those that just test for contamination organisms. So within the states, we do see this wide variety. So how is AOAC able to help in this place? Well, in 2018, AOAC established the CASP program. And AOAC essentially within CASP serves as this platform that can bring together stakeholders from various states to really try to help harmonize, the approaches and provide guidance to the states on how they could potentially update their regulations, what type of testing is being done, what data is being collected. So not only just for the microbial community, but as you can see, there's 5 different working groups within CASP that focus each on different targets. So while the presentation today, again, will be focused on micro, the CASP program in and of itself is really designed to provide a 360 approach to assisting the cannabis industry. So outside of this federal framework, the next main issue is standardization. So while we see different states have different regulations, there's also different approaches to how they are requiring or what their requirements are for the methods that would be used in those states. So within the CASP program, the microbial contaminants working group can develop what we call standard method performance requirements. And these SMPRs are really consensus standards that provide additional guidance to method developers or labs that are developing their own methods on how they can really validate their technologies that follows that robust peer-approved process that AOAC employs in other industries. And the Microbial Contaminants Working Group has done a fantastic job in the 2 years that CASP has been in place. There's been 3 SMPRs that have been developed, all for pathogenic organisms, so Aspergillus, STAC, and Salmonella, and allows method developers to then certify their methods using these SMPRs and then AOAC validation guidance in general. And then recently, the Microbial Contaminants Working Group has been focusing on an SMPR for quantifying yeast and mold in cannabis products as well. So these SMPRs, what do they cover? Well, they cover, validation guidance in general. So how do you need to prepare your strains? What sort of equilibration, stabilization periods are needed? How do we categorize the matrices? And we'll discuss a little bit about that more on the next slide. Are there specific, inclusivity and exclusivity? So target and non-target organisms that we should have that are more commonly found in the cannabis plant and should be included in our panels. How do we do culture confirmation? Other industries have established, cultural reference guidance, the FDA's BAM, USDA FSIS says MLG. We have different compendiums for water and wastewater and dairy products, but we don't have a global established, certified, collaboratively evaluated reference method for cannabis products. So the SMPRs have been designed to provide guidance on that so we can standardize that process as we certify methods. So as I mentioned, one of the key aspects we feel that the SMPRs have developed is really to identify different categories and essentially establish categories within the cannabis industry so that we can look at methods in general to see how they perform within certain matrices or within a category as a whole. We also work to establish specific test portion sizes that give us more robust data as we collect it during certification studies, and can really tell us more about the repeatability of methods and their ability to detect or enumerate targets. And there were, each of the micro SMPRs has identified 4 categories: plants, concentrates, infused edibles, and infused non-edibles. And the test portion sizes are consistent throughout all of these SMPRs. So AOAC brings everyone together. We then establish these SMPRs, and then we look to certify methods against these SMPRs, and then using AOAC validation guidance. And so the certification process has typically gone through the performance tested method program, which is AOAC, one of AOAC's 2 certification programs, and it has traditionally been used for methods that are sold as test kits or assays. And it is one of our premier certification programs. There's over 300 certified methods through the PTM program currently. And a lot of people that are coming into the cannabis industry aren't as familiar with the PTM program. So I encourage you to go to the AOAC website. There's the AOAC Research Institute and Performance Tested Methods program tab, and it provides a lot of information about these methods that are available. And you can then actually search in our database what methods are approved for cannabis. So you would just click on the link that's shown on your screen here to go to the PTM validated methods, and then you can search by matrix and type in cannabis or hemp and see which methods are certified. And briefly, we do have a few methods that are currently certified. So on the left-hand side of the screen, you'll see the GeneUp Salmonella 2 and the GeneUp EHEC method. Each of those has been certified through the PTM program and against the SMPRs that were developed for those targets. I also wanted to share, we do have several Aspergillus methods that are in various stages of the certification program, and we hope to have these out to our end users relatively soon, because it's very important to have certified methods for the end users. And then just not only what's on the screen here, but we have several methods. We have an expert emergency response validation going on for total yeast and mold, where we hope to have a minimum 7 methods certified to be used in the industry, as well as several other companies that are looking at these target pathogens or other potentially contamination organisms. So we really are encouraging our method developers that have traditionally focused on the food industry to transition into the cannabis industry, as well as encouraging the method developers that you can see on the screen that are currently in the cannabis industry to participate within our certification program. So one of the key challenges that the cannabis industry has faced is, sorry about that, we had a little bit of a blip in the recording process. But so one of the key challenges facing our community is this need for reference materials. So AOAC can certify methods, but we need our end users to verify that they're fit for purpose within their laboratories. And the easiest way to do that is through certified reference materials. So within the CASP program, we actually have So within CASP, we have this PT program that is looking to develop reference materials for both the chemistry side and the microbial side. And as you can see, it's chaired by Brent Wilson from NIST. And as I said, they're not only just developing cannabis reference materials, but they're also developing a QAP program, which is really a way where labs can evaluate the same set of materials, and then NIST can help determine, how close they are to the average results for all the participants in that. And then obviously outside of the proficiency testing program, there are some other PTs available. So they're great resource for the community, especially when you're looking to do verification testing. So one of the things obviously that we're doing today is education and training. And so we have our training and education working group that has put on this CASP week, which we're really proud of. In addition to that, there's several webinars that AOAC has put out through Cannabis Science and Technology or other potential resources, focusing on, how we develop some of these SMPRs, and then once they're developed, how you go about certifying methods according to that, so that laboratories know what AOAC is doing and what method developers are doing to really show that their methods are fit for purpose in the industry. Additionally, we have a lot of partnerships that we're looking to happen within the CASP program. We have one recently with our Hemp and Cannabinoid Science Institute that we signed with them to do some collaboration and research to get more information and educational materials out to the community, as well as AOAC's AOAC and Technical Division for Laboratory Management. These groups are within AOAC in general, and they can assist from a technical aspect, laboratory accreditation programs and those sorts of things. And I'd be remiss if I didn't mention that AOAC does put on workshops, and it's a fantastic training workshop that is focused on the AOAC process for certification and validation, but also contains specific training modules on method verification. So not only is this beneficial for test kit providers, so how they validate their method, but also then for laboratories on how they can go about verifying that those methods are fit for purpose. We also have some very specific cannabis SMPR discussion modules that we talk about the SMPRs and how they play a role within the community. And finally, the last, I put out there as a challenge is what future trends, and this is where AOAC is uniquely positioned to be adaptable to really figure out what could be going out, whether that is, doing more state and federal outreach to the Cannabis Regulators Association, USDA. How can we go out and partner with these groups to serve as a platform where we can either collect data for them, have our network collect data, and that sort of intersharing to really build better science. We also have emergency response validation. So I briefly mentioned earlier, we are currently having one go on for total yeast and mold, where we're evaluating several methods using a singular set of data to really get test methods out to the community in a quicker process. And we're keenly set up to do this for other targets as needed for the industry. We can also do targeted matrix validations where we actually focus specifically on specific matrices that may be causing issues in the community. So say maybe it's a cannabis-infused beverages or cannabis infused therapeutic patches, and there's outbreaks associated with them where we can then take certified methods and really evaluate them for that specific matrix in a pretty quick process. We're also positioned to look at new analytes. So not a lot of states have guidance for, say, listeria. And so we can provide guidance on how methods could be evaluated to detect listeria, which is found in almost any production facility, as well as anything else that emerges and could become a problem for this industry. So thank you again for your time today, and I really appreciate you listening to this presentation, and hopefully we can get you to join our microbial contaminants working group.
Thank you. First, thank you all to the speakers. That was a great session. I learned a lot. I had some great questions come in on the chat. So I'm going to go to that, and these aren't addressed to anybody in particular. So if one of the speakers could raise a hand or jump in, I'd appreciate that. So just starting from the top, we have some questions around what pre-treatment do you recommend to do to an oil extract and a CBD crystal? Who would like to field that one first?
Toby, I can kind of start with that.
Thanks, Alex.
Yeah, pre-treatment is a little bit interesting because of the variation that we see in matrices, but for concentrate samples, we'll use a surfactant in the enrichment broth to kind of help dissolve some of that kind of sticky, thick material so that it is more easily enriched and able to be analyzed.
Thank you. Following on from that, there was a question about homogenizing dry cannabis, cannabis oil extract, and distillate. Could you expand on that? And then maybe one of the others could add anything else?
Absolutely. So alluding to what I had mentioned in my talk about small sample sizes, we are dealing with small sample sizes. So we find that it's easiest to hand massage instead of putting a small bag in a stomacher or something similar. So hand massaging to thoroughly mix samples. And I see Mike Esposito nodding, so I'll let him take over.
Thanks. Mike, anything you'd like to add?
Absolutely. I would second the hand massaging as well. We oftentimes see as well, you'll run into materials that might have some sharp edges like hard candies or some thick stems on a cannabis plant. So you want to kind of be aware of that as you're homogenizing to make sure you're not producing leaks or punctures or anything like that. Then also naturally, you want to try to kind of break the material down a little bit before you homogenize. We know a lot of these microorganisms are deep inside the inflorescence here, so you want to be sure that whatever liquid you're applying to that's able to really get in there and liberate anything so that you're able to actually assess it.
Awesome, thank you. Pat, Michael, would you like to add anything on that topic? Okay, moving on to the next one. We have a number of questions around USP. I'll summarize a couple and then let the panelists speak up. Someone's asking, is there a reference method for cannabis from the USP? And secondly, given the pharmacological activity of cannabis, is it considered a medicine? Taking this into account, would the USP microbial criteria apply for it? Michael, I'll look to you first because you gave a great summary of the regulatory agencies around cannabis. And then please, the other panelists would like to share any insight they may have around the USP connection.
Sure. So this is where it gets tricky, right? It depends on what your product is, right? If you're selling a topical or some kind of dietary supplement or something that is, typically would fall under FDA jurisdiction, they're going to look to USP reference methods as the reference. I can't remember what you were asking specifically about. I'm trying to scroll up to the actual question. Does USP 1111 acceptance criteria apply? Again, USP doesn't really take the approach of very specific matrices. Rather, they look at general types of products, and then you are expected to do a risk assessment to determine, A, does my product most closely fit this kind of product? And then even in 1111, right, it's a general guidance chapter. So the acceptance criteria there are recommendations, minimum requirements if you want. It's still expected that you would do a risk assessment to determine, do these acceptance criteria make sense? Are there other bugs that I need to test for as part of my qualitative analyses? So when you're getting into USP, a lot more responsibility is placed on the manufacturer to risk assess what needs to be tested for.
Great, thank you, Michael. Would anybody else like to add to that?
Toby, I'd like to just kind of add on to that. I think the USP in general is a fantastic guidance document, and we actually took that into account within the microbial contaminants community as we started developing our standard method performance requirements for these different organisms. So we tried to align test portion sizes with those recommended in there. And a lot of the confirmatory processes that we have established in the SMPRs do align with both the FDA and USP to some extent. But I really do think when you're looking at it, to add on to what Michael said about general kind of guidance, you should look to the state. So the states have their action limits for, and they have various organisms that are listed. So it's going to vary. So just following USP is not going to be sufficient for testing a product. You really do need to look to see what does your state require? What are the acceptable action limits for that? And from my experience, Mike Esposito did a fantastic job talking about the culture-based methods. Lots of labs though are using more traditional rapid methods. So whether that's a plating-based method that's like a film-based or PCR or some sort of molecular detection. So there's not necessarily a lot of methods or a lot of companies running USP-based cultural methods for analysis. To my understanding, it's much more rapid technology.
Thanks, Pat. I'm going to keep you on the line there, Pat. And could you maybe speak to the question? I think it might be related or might not be, but the question is, where should labs look to get an ongoing method performance QA, QC requirements and criteria? Once labs verify the method initially using an SMPR, it seems like ongoing performance is lacking. Could you speak to that?
Absolutely. So the SMPR, it's very interesting. SMPRs for the micro community broadly are developed for method developers. And the idea behind the SMPR is to have a singular organization, a company, certify and validate their method through AOAC. And then laboratories would be able to onboard that technology through some sort of verification, PTs, initial spike samples, or something to that extent. Any of the methods you're running, you should be running quality control strains routinely with those. So whether you're batching them daily or multiple runs within a day, you should be having positive and negative controls, maybe even sterility controls that you would run through there. And then annual performance in PTs, ring trial studies. So we don't get into as much, say, like suitability requirements on the micro side as they do in the chemistry SMPRs. But there is, just general guidance for running, quality controls and PTs that would be outlined in 17025 accreditation that you should look at.
Thank you, Pat. Appreciate that. Would anybody else like to add any comments? I covered it. Perfect. There's a couple of technical questions that I'm going to throw out to the panel, and please just chime in. First one is, how does the microbial load vary throughout the 4 matrix categories? Are there trends we see across the sample types? Mike Esposito, would you like to start there?
Yeah, absolutely. I will say, having worked in the production aspect of this before moving on to the laboratory side, I would say, in my experience, the highest bioburden is absolutely going to be on the flower material for a multitude of reasons there. Number one being it's an agricultural process, so naturally you're going to have some microbial presence there. Number two being a lot of cultivators. In a world where you're not confined by regulations, you're going to be adding a lot of microbial inoculants to your plant products. You might be adding some fungus and bacteria to your root zone, and that's extremely easy for you to accidentally transport up onto your flower material. And then once that's kind of in there, once that's in your HVAC systems, your drying rooms, it's much harder to eradicate that. So I would say, in my experience, the flower matrix has the highest bioburden by far.
Great, thank you. Would anybody else like to add to the sample diversity or the loads? Alex?
I will absolutely echo what Mike said. I think flower is the biggest concern. The inflorescence is dense. And I think in large part too, what we see potentially could be people hand packaging and hand trimming who maybe don't have as good of hygiene practices or glove use, things like that, sample to sample, plant to plant too.
Thank you. Great. And Alex, I'll keep you on the mic. Can you answer the question, how much tween 80 can be added for oily samples to facilitate dissolution?
That's a tricky question, and is sort of left to internal validations and verifications that labs can perform. We see anywhere from 0.1 kind of to 1% tween in some instances, and I think I've even heard of people going higher than that. But really, it's what the lab is comfortable with in terms of their spike and recovery studies for the tween use in concentrates and some of those oily topicals.
Great, thank you. And I assume the panel would agree with that as well. Awesome. Mike Esposito, can I ask, how does your lab handle presumptive positive results for pathogens? Is a confirmation performed or is action taken on the preliminary results?
All right, that's a great question. We always want to have a secondary cultural confirmation if we're picking up a pathogen on our microbiological assays. There's a few ways you can approach that. A lot of labs will validate in-house a culture-based method. Our lab, we actually have a secondary lab that's validated for culture-based methods that we would send our sample extract out to. They would perform a culture-based confirmation. That way we can either rule out, was this a false positive or is this a true hit?
Great, thank you. Michael, Alex, would you support, differ to that?
Cultural confirmation on a presumptive is, I think, pretty robust, and that's what we also do, a secondary confirmation, just to make sure that what we're calling is actually a positive. Great, thank you. Michael.
Yeah, I would just add that the lab is not going to summarily take action on behalf of the client, or we're going to notify the client of the results and see what they want to do. I'll also add that there are alternatives to cultural confirmations that have gained traction in the food industry, and I expect they're going to gain traction in the cannabis industry as well. There's the Bruker-Malditoff MSID, some people call it, mass spec-based identification systems. There's others that are coming to market as well that can get you, in some cases, a quicker time to result in the cultural confirmations.
Great, thank you. And Michael, I'll keep you on the line. There was a question around some components of cannabis inhibit PCR. Can either of the mics take that question?
Sure, I'll jump in and Mike can finish off. I mean, yes, I'm not sure what the question is, other than yes, you're correct. There are lots of inhibitors, and it's like there are in any complex matrix. But I mean, there's some specific ones that are common in the cannabis plants, where there's polyphenols that inhibit the amplification. There's a lot of these, there's edible type products that you're adding a lot of sugar that sequesters DNA, makes it difficult. There's metals, magnesium and other chelators that are going to inhibit binding of the primers. There's all sorts of things that can inhibit the PCR reaction, which is why it's so critical to do your inline controls, as Pat was saying, and why it's so critical, as we can't remember which, several of the presentations I think mentioned it, to do some kind of methods or matrix-specific suitability if you're going to go the rapid path.
Great, thank you, Michael. Mike, would you like to add anything there?
Yeah, absolutely. I just wanted to second all of that, and then I also kind of wanted to note how that sort of highlights the importance of an enrichment step. When you're dealing with these matrices, you really need to make sure your target is available for detection there. Along with the endogenous compounds in the cannabis flower that could inhibit, you also have to keep in mind that you're not really ever aware of what your client is applying to this sample. So if you're using a molecular method, you really want to be sure that you have like an internal control curve there that's going to really show you if you have the inhibition. Another thing to keep in mind if you're trying to do any sort of CQ to CFU conversion there is anything that's going to push your CQ back a few cycles is going to really throw off your CFU result in the long run there. But with the more detection-based ones, you can kind of circumnavigate this by doing like a dilution of your extracted sample, secondary enrichments, etc. There's a few ways you could approach that.
Thank you, Mike. That's great insight from both of you. Appreciate it. Interesting question just popped up, and maybe the panel can comment. As pooling validations become popular for COVID testing, do you see pooling for PCR detection becoming common in this industry? comments. Pat, maybe.
Yeah, I'll jump in on this because we actually spoke a little bit to this comment last week on our CASP call. So pooling can really mean two different things. We can do wet pooling, where we're taking post-enriched samples and combining that together, as well as pooling dried samples or just the product itself, compositing them together and then doing an enrichment on it. I do think both will be a part of the cannabis industry in the future. We've seen that trend in the food industry, explode over the last decade. Ensuring that the methods are robust enough to be able to detect a single positive sample and a compositive sample, whether that's in the dry or the pooling stage, I think is the key. So it takes us refining the not only the detection tech part of it, the enrichment portion of it, any extraction that would be there. So it is going to happen, and it wouldn't surprise me in the next couple of years if we start seeing it being more common.
Thanks, Pat. Great insight. Would anybody else like to add anything to that? Okay. Last question, maybe. To carry out the validation of a microbial method in the cannabis matrix, is it necessary to do it by category? I know some may have touched on earlier, but the short answer from someone would be?
Short answer is yes.
Yeah, okay. And I think that's one of the challenges we see with the categories continually evolving in the marketplace. Maybe one last question, and this isn't just towards the microbial discussion, but generally to the entire cannabis ecosystem. Lab shopping is a topic that happens. From your perspective, maybe with a microbial focus, could you share your insight on how we collaborate or work together to minimize this and ensure that the right data is making it to the regulatory agencies? Alex, would you like to start?
Yes, thank you. So Toby, in my opinion, I think the first step towards avoiding that is changing the mentality around testing, which I think stems primarily from education and demystifying the science of what we do. I know I talked about clients not understanding, and there's opportunities, I think, for us as labs to become more of an outreach kind of component in the cannabis space and work more together collaboratively as labs, even within states or across state lines, just to kind of become a resource for education, so that mentality maybe shifts away and we become more of a resource to explain and understand. And we can answer things like, what are these quality systems that you use, and what does this data mean, and what is an inline control? Because I don't think at this point a lot of folks understand that we really are trying to make our science as robust as it can possibly be.
Well said. Yeah. Michael, Mike, Pat, would you like to add anything to that response?
Yeah,
I think I would just add that. No, go ahead, Mike.
You go first.
All right. I would just say, I think that in our experience anyway, it's helping to consult or, with the clients has been a huge way to make them feel more comfortable with the result. Is that, okay, you got this result you weren't expecting, none of us are happy about it, but here's what we can do to help you try and root out where did it come from, what does this mean for you, what can you do next. Otherwise, if you're just throwing them a result, it's, okay, your material's out of spec, then they don't know what to do with that and they get frustrated. So I think that the lab is well placed to help explain the result, as Alex said, and then, what are some next steps that you can take? And CASP is going to be a wealth of knowledge for that sort of thing, because we're bringing together method developers, labs, and then the production companies. So we're going to get to see through CASP what are the common root causes, because these things are common across the board a lot of times.
Thank you, Michael. Mike, would you like to add anything?
Yeah, definitely. I want to second what both Alex and Mike said there. 100% for us, speaking with our clients and trying to help them get to the bottom of their problem, like Mike said, is huge. And then aside from that, it's just on the labs to be very responsible when they're selecting technologies, making sure there's incredibly robust validation packages. And then it's also on the state regulators as well. If you have labs that have a competitive advantage because of extremely high turnaround or low fail rates, you do need to investigate that a little bit. But really, it comes down to what both of the previous speakers said. We should just be communicating with our clients as openly as possible.
Awesome. So I think we're unfortunately out of time, so I'm going to have to stop us there. But I just want to say a huge thank you to all of you for presenting today. Thank you to the audience for tuning in. We look forward to bringing you more initiatives on microbial testing in the CASP network in the year. And please tune in tomorrow for the final webinar on chemical contaminants. Until then, everyone stay safe and thank you.